Hi, welcome back. Today we're going to cover Chapter 7, Microbial Biochemistry. We'll focus on organic molecules and carbohydrates, and these are the chemical building blocks of life, and understanding them will help us connect basic chemistry to microbiology and human health. Chapter 7 is divided into 5 sections. We'll notice that we will cover carbs, lipids, and proteins. Nucleic acids are also a type of organic molecule, but we're going to cover that further down the road in Chapter 10. Section 7.1 will give an intro or overview of organic molecules, and by the end of this section, you should be able to identify common elements in organic molecules, explain isomerism, recognize functional groups, and describe how these groups form polymers. Back in the 1950s, Stanley Miller and Harold Urey asked, could organic molecules form from simple chemicals that were present in Earth's early atmosphere? So what they did was simulate lightning on gases like methane, ammonia, and water vapor, and the experiment produced amino acids, which are the building blocks of proteins. This showed that the chemistry of life could arise naturally from simple conditions. Living cells are mostly made of just a few elements, including carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And together, these account for about 99% of the cell's dry weight. We call these macronutrients. Trace elements like iron, zinc, magnesium, and others are still needed in smaller amounts, but are still essential, and these are called micronutrients. The 4 most abundant elements in living matter are carbon, nitrogen, oxygen, and hydrogen. And I often just think of like my last name, Chen, except with an O. So Chan. Chan makes up the 4 most abundant elements, and they also happen to form very strong covalent bonds. Because of their ability to form bonds with one another, they form very diverse molecular structures, which is the topic for this chapter. Molecules can be subdivided into 2 types. Organic molecules always contain both carbon and hydrogen. Examples include sugars, proteins, and fats. Inorganic molecules, on the other hand, include things like water, salts, or CO2, and they do not contain both carbon and hydrogen together. While organic molecules form the structures of life, inorganic molecules are smaller, more simple, and support cell function. We'll also see in this chapter that these carbon skeletons can be formed from organic molecules when they're linked together in chains, branches, or rings, and these are going to form the building blocks of life. All right, so we saw the most abundant elements in living cells or in nature are hydrogen, carbon, oxygen, nitrogen, phosphorus, and sulfur. These are 6 macronutrients that account for about 99% of the dry weight of cells. There are other elements that are needed, like sodium, potassium, magnesium, calcium, iron, zinc, but these are needed in much smaller amounts, and we call these micronutrients or trace elements. For the second question, what are the differences between organic and inorganic molecules? We said organic molecules contain carbon bonded with hydrogen. They're usually larger, more complex, and they form the structures of living cells. Whereas inorganic molecules are small, simple, and they lack those carbon-hydrogen bonds. So examples include the 3 shown here: carbon dioxide, ammonia, and oxygen. Carbon is special because it forms 4 covalent bonds. This allows chains, branches, and rings of different shapes to form. Molecules with the same formula but different arrangements are called isomers. For example, glucose, galactose, and fructose all share the same molecular formula, C6H12 and O6, but they differ in their structure, right? So same molecular formula, but differ in their structure because they differ in how they are bonded together, their bonding sequence. Some isomers differ in their 3-dimensional arrangement, and they're called stereoisomers. A common type of stereoisomer are enantiomers. These are mirror images of each other, kind of like your left and your right hands. But these enantiomers are also chiral. That means they are non-superimposable. So what does superimposable mean? Superimposable means that two objects can be placed on top of each other, so they're not only mirror images, but they can be placed on top of each other in such a way that they completely overlap, aligning perfectly. Whereas non-superimposable, like the one up here, our hands, you cannot place them on top of each other. They are not going to perfectly fit. They do not align, although they are mirror images. Since enantiomers are chiral or non-superimposable, usually only one enantiomer, so one version, often fits into enzymes or receptors. And this is why chirality is so important in biology and medicine. And our book provides a few examples of enantiomers, these non-superimposable mirror images. One is sugar, the glucose, and here we have an amino acid, alanine. Certain species of mold, yeast, and bacteria can only metabolize one of the optical isomers, so one of them and not the other. The other would not be a suitable source of nutrients. Another reason enantiomers are important is because let's say you're using a drug as some kind of antimicrobial medicine to treat an infection. For these microbes, they might be affected, right? Maybe one of the drugs, one of the enantiomers can kill them, but the other does nothing to harm the bacterium or whatever microbe you're trying to kill. In further application to pharmaceutical drugs, drug companies often sell drugs as racemic mixtures. So these are when you have both enantiomers of a chemical together. And then later, when their original patent is close to expiring, they might market the single active enantiomer as the new and improved version. Another pharmaceutical application is the thalidomide disaster in the 1950s and 60s. So thalidomide was sold as a morning sickness drug, so to prevent or decrease the morning sickness symptoms in pregnant women. But it was sold as a racemic mixture, so both of the mirror images were in the drug. The R form is the one with the desired effect, the sedative effect, but the S form was teratogenic. So teratogenic means that it's a substance that can cause functional birth defects in the developing fetus. So unfortunately, this led to birth defects and miscarriages in the pregnant women who took this drug. And even taking just the R form would not be safe because it turned out that the enantiomer can interconvert. So they can convert from R to S in the body. So both forms would pose risks. The disaster did have an outcome that improved drug regulations over time and also awareness of how important it was to know which enantiomer was in the drug or was the drug made of, and also if the body could interconvert those enantiomers into a more harmful form. All right, so our checkpoint this time is about carbon. What makes it so suitable to be part of all the macromolecules of living organisms? It's really that carbon has those 4 valence electrons, which allow it to form 4 stable covalent bonds. And carbon can bond with many elements: hydrogen, oxygen, nitrogen, sulfur, phosphorus, and other carbons. So carbon atoms often link in chains, branches, or rings, producing an enormous variety of stable molecules of different shapes and sizes. So this versatility really makes carbon the backbone of all major biomolecules. Organic molecules often contain functional groups. These are small groups of atoms that determine how molecules behave, and they are categorized based on their specific chemical composition and the chemical reactions they perform. So examples include some groups you might have seen before in other classes. Hydroxyl groups, OH. We have carbonyl, which is C double bonded to O. We have carboxyl, carboxylic acid, as shown here, which kind of is a combination of both carbonyl and hydroxyl. Amino, we're going to see amino groups in our amino acids. Carboxyl groups are also in our amino acids. And phosphate groups you've probably also seen this one in your previous classes. These groups give molecules their chemical reactivity. Most biomolecules are polymers made from smaller monomers. For example, glucose monomers link together to form starch or glycogen. The process that links monomers is dehydration synthesis. Water is released when the bonds form. Breaking down polymers apart uses the opposite direction, called hydrolysis, and water has to be added to the reaction. In this chapter, we'll look at different macromolecule groups. There are actually 4: carbohydrates, which are used for energy and structure, lipids for membranes and energy storage, proteins, which are often enzymes and offer structural support, as well as nucleic acids like our DNA and RNA for genetic information. But this one we're going to look at in chapter 10. So this chapter will look at the first 3. So dehydration was the formation of polymers from smaller monomer units. And we can see one monomer here, one monomer here. When they're connected together, these are gone and water comes out. Water's released from the reaction. And we have our specific example in the book. We have glucose. Here's one glucose molecule. Here's another. And I can see that when they link together, when a covalent bond forms between the 2 monomers, I get water formed as a result of the reaction. Once you finish the entire chapter, I recommend coming back to this table and making sure you can name the types of macromolecules as well as their overall functions. Remember, we're going to talk more about nucleic acids in chapter 10. So that byproduct of dehydration synthesis, remember, was water, H2O. Water is released when monomers are joined to form polymers. All right, let's recap 7.1. Identify common elements and structures found in organic molecules. We talked about carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur as being the most common elements. Organic molecules are built on carbon skeletons. These are chains, branches, or rings, and these skeletons can attach to functional groups, giving molecules their specific properties. For the second one, the concept of isomerism. Isomers are molecules with the same chemical formula, but different structures. So we had structural isomers that differed in their bonding arrangements, like glucose, fructose, and galactose, and stereoisomers that differ in their 3-dimensional orientation, like those enantiomers that are mirror images of each other. Even small structural differences can really change their physical and chemical properties, as well as their biological function. For examples of functional groups, we talked about a few: hydroxyl, the OH group, carbonyl, the COO, and we're going to see later those are found in things like aldehydes and ketones. Carboxyl groups, COOH, we'll see that in amino acids. Amino groups, the NH2 is also found in amino acids. And phosphate groups. The last one, the role of functional groups in synthesizing polymers. So we talked about how functional groups determine how monomers interact and bond. In dehydration synthesis, functional groups like hydroxyl and carboxyl might react, releasing water. So water is a product of the reaction, and this enables the formation of covalent bonds between monomers, building these larger polymers like carbohydrates, proteins, or nucleic acids. All right, our next section is 7.2, carbohydrates. And by the end of this section, you'll be able to give examples of monosaccharides and polysaccharides and describe their functions in the cell. Carbohydrates have the general formula CH2O with N over here, so that if N, for example, were 6, this would be C6H12O6, such as we saw in glucose earlier. They're often called hydrated carbons because it looks like you're adding water to carbon, right? You're attaching those together. So this is really what led to the term, the naming of carbohydrates. It's like hydrated carbon. Carbohydrates provide energy storage, like glucose, starch, or glycogen. They can provide structural support, like the cell walls that we've seen in previous chapters, made of cellulose, chitin, or peptidoglycan, and they can even form part of RNA and DNA backbones. In biochemistry, carbohydrates are often called saccharides from the Greek word meaning sugar, although not all saccharides are sweet. Monosaccharides are the simplest sugars. These are single units that are named by their carbon number. If they have 3, this is triose. 5 is pentose, 6 is hexose. So examples include glucose, which is the main fuel for cells, galactose, which when combined with glucose forms lactose, and fructose, which is fruit sugar. In water, many of these monosaccharides will form stable ring structures like glucose. Here we can see the 3 I mentioned earlier with 3, 5, and 6 carbons respectively. And then we have aldose on the far left with an aldehyde group, and a ketose here with a ketone group. Monosaccharides with 4 or more carbon atoms are often more stable when they actually adopt a ring or cyclic structure. These ring structures form from a chemical reaction between the functional groups on opposite ends of the sugar's flexible carbon chain. So here, for example, the carbonyl group and the hydroxyl group are going to react together to form a ring structure. Glucose, for example, on this far right shows or forms a 6-membered ring. So again, why do they form ring structures? They have this flexible chain, and the functional groups at the ends of the chain will react, forming a stable cyclic ring structure, which is actually more energetically favorable than a straight chain. Disaccharides, where di refers to 2, are just 2 monosaccharides joined by a glycosidic bond. So glycosidic bonds are what form between monosaccharides to form these multiple saccharides linked together, like disaccharides, oligo, or polysaccharides. Here, they're formed using that dehydration synthesis we saw earlier. You can take monomers and connect them together. Here we're going to form glycosidic bonds, and water will come out of the reaction. So it's not shown here, but you would see water is a product of the reaction. Examples of disaccharides include maltose, which are 2 glucose molecules linked together, lactose, which is galactose and glucose, and sucrose, which is glucose and fructose. Oligosaccharides are a few more linked together. So there are usually around 3 to 10 monosaccharides linked together using those glycosidic bonds, and they often function in cell recognition and signaling, especially when attached to proteins or lipids, and we'll see them called glycoproteins or glycolipids when they're in that form. And here's another look at those disaccharides. We have maltose, lactose, and sucrose with those glycosidic bonds connecting the monomers, and these were formed through dehydration synthesis. Polysaccharides are large polymers of sugars. Usually they're not sweet, and they're also insoluble, not soluble in water. These include starch, which is often used as energy storage in plants, glycogen, which is how we store our energy, so we're humans or animals. Bacteria can also store energy in the form of glycogen. Cellulose, we saw these in plant cell walls earlier, so they have a structural function. Chitin, these make up the cell walls in fungi, and actually also make up the exoskeletons in insects. And peptidoglycan, we saw this earlier in bacterial cell walls. So 3 of the most common polysaccharides are shown in this picture: starch, glycogen, and cellulose. What's really interesting is they're all made of the monomer glucose, but they use different bond types to connect the glucose monomers together. And the orientation of glycosidic linkages in these 3 polymers really, because they're different, they have consequences. They have different properties. Humans, for instance, can digest starch. We can actually break the bonds between the glucose monomers. We can also do this in glycogen to release individual glucose molecules, but we cannot digest cellulose, which is also known as fiber. And because of that, it stays in the large intestine, attracting water through osmosis, and allows it, or it makes it easier for us to go to the restroom. All right, so we talked about many important polysaccharides. Starch, that main energy storage in plants. Glycogen, the main energy storage in us, animals and bacteria. Cellulose, we saw that as structural support in plant cell walls. Chitin, those were in fungal cell walls and in insect exoskeletons. Peptidoglycan in bacterial cell walls as a structural component. So these polysaccharides are really critical for energy storage and structural integrity across many different types of organisms. All right, let's recap carbohydrates. For the first one, examples of mono and polysaccharides. So monosaccharides are our simple sugars like glucose, galactose, fructose, ribose. For polysaccharides, these are large monomers, like hundreds of monosaccharides linked together, including starch, glycogen, cellulose, chitin, peptidoglycan, and others. For the second question, describe the function of monosaccharides and polysaccharides within a cell. So monosaccharides serve as immediate energy sources, like glucose in cellular respiration, which we're going to talk about soon. They act as building blocks for larger carbohydrates as well. Polysaccharides store energy, like starch in plants or glycogen in humans, and they provide structural support in the cell walls, for instance. All right, so that takes us to the end of 7.1 and 7.2. In the next section, we'll begin with lipids. Thanks for watching.